All right, so we have talked about the blood grouping system, that is the ABO blood grouping, and now we are going to dive into the RH blood grouping. And this RH blood grouping again is determined by the presence or absence of a factor which is called the RH antigen, which is again present on the surface of the RBCs. And this RH antigen is very similar to the ones that are found in the rhesus monkey. That is why the name is derived from the rhesus monkey, RH. And if a person is having the RH antigen on the surface of the RBC, he or she would be RH positive. If not, it would be RH negative. So as simple as that. Now, RH negative person, if exposed to RH positive blood, then they will produce antibodies against RH antigen, only if exposed to the RH positive blood. Now here comes a special case which is called the erythroblastosis fetalis. This is a disease which is caused by RH incompatibility in a pregnant mother who is RH negative. So the mother is RH negative and the fetus is RH positive. Now what happens when the fetus is in the womb, then there is a clear separation of the blood, and this is done by the placenta. So there is a clear separation, and that is why the two bloods of the fetus and the mother, they do not mix. So no mixing. Now this is the case of the first pregnancy. If the first pregnancy has a fetus to be RH positive and the mother is RH negative, then the placenta is separating the two bloods and the bloods are not mixing, thereby there is no problem. But during the delivery, when the first baby is delivered, there is a little mixing of the bloods, and since there's a little mixing, the RH negative mother would be exposed to the RH positive blood, and hence the RH negative mother would develop antibodies for RH antigen. But there won't be any complications with the child or so. The complication happens during the second pregnancy. And if the second pregnancy is giving a child who is again RH positive, so RH positive, second child, right? So the second pregnancy also has a child who is RH positive. Now, during the second pregnancy, when the child is inside the mother's womb, there can be some leakage. There can be some leakage of blood from mother to the child. And because of the first pregnancy, since the blood is having the antibodies against the RH antigen, this is harmful for the fetus. Now, since there is a leakage of blood, this is going to destroy the RBC of the fetus. Now, this is the complication. And this destroying of the RBC, this can lead to severe anemia and also jaundice. Right? So because the blood is leaking and the blood is already having the antibodies for the Rh antigen, that is why the second child can have some severe conditions like anemia. So this can be the case. So to avoid this, immediately after the delivery of the first, child, the mother who is having the Rh negative and the mother who is actually the Rh negative mother and is now having antibodies against the Rh determinant or the Rh antigen, some anti-Rh antibodies, these antibodies are administered to the mother, because the antibodies that are present, the antibodies that have developed because of the exposure to the Rh positive blood, that can be neutralized by the anti-Rh antibodies. Let's say this is the antibody that is developed against the Rh positive blood. So this is the Rh antibody. Now this antibody is going to be neutralized by another antibody, which is the anti-Rh antibody. So that is why the mother is administered with this anti-Rh antibody. And that can bring the condition to a stable condition and the complications can be avoided. So we're going to continue from here in the next video, and we are going to look at the blood clotting or blood coagulation. We're going to look at the steps and the different molecules or different proteins that are involved in blood clotting or blood coagulation.